We decided to participate in the Lorentz workshop Life Science with Industry because I liked the idea and I was curious to see with which kind of ideas and surprises the PhD students would come up.
At the end of my PhD, I participated in the workshop Life Science with Industry. I signed up for this workshop because it seemed like a good opportunity to interact with industry, since I would be looking for a job soon when I finish my PhD. I chose the BEO project because it was about breeding. I did my PhD on plant biology, but I didn't have any experience with breeding, and it seemed like a good idea to get some hands-on with a breeding project to see if I would like to work in this field, but also to get some insight in what is going on in the breeding industry.
For our industry, knowledge and technology is becoming more and more important. A new application within this field is genomic selection. And the idea behind this new method is simple. You use, let's say, 200 plants to genotype and to phenotype, and with this data you use statistics for another population of, let's say, 2,000 plants to select the best plants from this population.
BEO asked us to investigate if genomic selection can be used for breeding vegetables like carrots or cabbage faster and more efficient at Beozade. And the first day we made us familiar with the topic breeding and genomic selection, and soon we realized that the question was too broad to give a ready-made answer for Beozade. So we discussed with Henk if we could change the project a bit in coming up with recommendations for things that are necessary to actually even start thinking about doing genomic selection at Beozade. We divided this in three subjects that needed to be in place before genomic selection can be implemented. This is phenotyping, developing a statistical model, and genotyping, so looking at the DNA of the crops. For phenotyping, for example, we came up with an idea to make photographs of the carrots to phenotype them instead of looking at them by eye. With this, you can come up with a quantitative number for traits like, for example, carrot size, length, color, all kind of things. You can even use these pictures later on if you want to investigate a different trait.
At the start of the workshop, the students were not familiar with breeding. Fundamental research and breeding are far apart. But after my introduction about breeding and genomic selection, the students started to ask many questions. I was surprised by the inventiveness and the enthusiasm of the students. So my conclusion was that we shouldn't start with genomic selection overnight, but take our time to set the conditions right to make this successful.